Hello everyone, welcome to a new session of the paper Molecular Biology. In this session, we will discuss translation regulation, RNA instability and inhibitors of translation. Translation is the process of converting the genetic information encoded on mRNA to amino acid sequences. The overall process is divided into three stages called initiation, elongation and termination. Each step differs in both prokaryotes and eukaryotes. Gene expression is regulated at multiple levels, including the translation of mRNAs into proteins. Compared to transcriptional regulation, translational control of existing mRNAs leads to more rapid changes in the concentrations of the encoded proteins in the cells. Thus, translation regulation can be used for maintaining homeostasis in addition to modulating more permanent changes in cell physiology. Translation may be controlled by regulators that interact with mRNA. The regulatory products may be proteins, which often are controlled by allosteric interactions in response to the environment, or RNAs, which function by base pairing with the target RNA to change its secondary structure. Regulatory networks can be created by linking regulators so that the production or activity of one regulator is controlled by another. General features of translation. The process of translation can be divided into initiation, elongation, termination or ribosome recycling. Requirements for translation: 1. Amino acids. 2. Transfer RNA or tRNA. In this, you have amino acid attachment site and anticodon. 3. Amino acyl tRNA synthetase. 4. Messenger RNA or mRNA. 5. Functionally competent ribosome. 6. Protein factors. 7. ATP and GTP required as a source of energy. Translation regulation strategies. Mechanisms of regulating translational initiation fall into two broad categories: those that impact on the EIFs or ribosomes and therefore affect virtually all scanning dependent initiation events; those that impact on the mRNA itself either through sequence specific RNA binding proteins or microRNAs or miRNAs and are therefore potentially selective for certain mRNAs. Most regulation is exerted at the first stage, where the AUG start codon is identified and decoded by the methionine tRNA specialized for initiation. The mechanism of start codon selection differs fundamentally between bacteria and eukaryotes. Accordingly, the strategies for regulating initiation also differ. In bacteria, base pairing of the Shine-Dalgarno or SD sequence located just upstream of the start codon with the complementary anti-SD sequence in 16S rRNA recruits the 30S ribosomal subunit directly to the initiation region of the mRNA. Hence, most translational control in prokaryotes involves modulating the mechanisms to access the SD sequence, whereas in eukaryotes the start codon is generally identified by a scanning mechanism where the small 40S ribosomal subunit loaded with methionine tRNAi in a pre-initiation complex or PIC binds to the mRNA near the 5' end and scans the 5' untranslated region or 5' UTR for an AUG codon. Consequently, RNA structures that slow down the ability of ribosomes to interact with the 5' UTR in single stranded form or to scan the 5' UTR reduce the efficiency of initiation. Decoy AUG codons in the 5' UTR can also intercept scanning ribosomes to hinder recognition of the correct start codon. Mechanism of initiation regulation. 1. Cap dependent initiation regulation. The major steps involved in the mechanism are as follows: formation of 43S pre-initiation complex, attachment of 43S complex to mRNA, ribosome scanning of mRNA 5' UTRs, initiation codon recognition, commitment of ribosomes to a start codon, ribosomal subunit joining and reinitiation. Most of the regulation occurs at the initiation stage of translation. The initiation pathway is comprised of several reactions that allow the binding of the AUG start codon of mRNA in the P site of the ribosome base paired with the anticodon of met-tRNAI. As said before, the start codon is identified by the scanning mechanism. A 43S pre-initiation complex comprising of the methionine tRNAI and initiation factors EIF1, 1A, 2, 3 and 5 is allowed to bind to the 5' cap of mRNA, which is facilitated by the cap binding factor EIF4E and EIF4A in the EIF4F complex. The PIC then scans the mRNA downstream, checking the codons as they enter the P site, complementary to the anticodon of methionine tRNAI. The methionine tRNAI bound to the PIC by the GTP linked EIF2 when reaches the AUG start codon stops scanning and triggers the irreversible hydrolysis of GTP in the EIF2 GTP methionine tRNAI ternary complex TC. The large 6Ts subunit joins to form an ATS initiation complex ready to accept the appropriate amino acyl tRNA into the amino acyl site and synthesize the first peptide bond after the release of EIF2 GDP and other EIFs. The initiation factor EIF4E recognizes the 5' cap and promotes the recruitment of EIF4G, generating the initiation complex. Ribosome is recruited at the 5' end of mRNA and translation takes place once the scanning of 5' UTR is complete and the AUG or start codon is reached. Factors regulating start codon selection. Start codon selection is regulated by various factors. Changes in the action of the initiation factors can affect the whole process of translation initiation. EIF1 collaborates with EIF1A and promotes scanning from the 5' end of the UTR through near cognate start codons, which occurs on an unstructured mRNA without ATP hydrolysis by EIF4A, the helicase subunit of EIF4F. The EIF1 stabilizes an open conformation of the mRNA binding site of the 40S subunit by modulating the conformation of the mRNA entry channel and promotes scanning when non-AUG or start codons occupy the P site. EIF1 also discards non-AUG codons during scanning by blocking the release of PI or inorganic phosphate from the partially hydrolyzed EIF2 GDP PI in the scanning PIC. EIF1 dissociates from its location near the P site in order to neutralize these gatekeeper functions at the AUG codon. Consistently, EIF1 mutations increase initiations at near cognate start codons by allowing premature EIF1 dissociation, whereas overexpressing EIF1 suppresses UUG initiation in certain mutants. The EIF1A thought to occupy the A site also regulates start codon selection. Their C-terminal region promotes continued scanning at non-start codons or AUG codons, whereas N-terminal arrest scanning and promote EIF1 release at AUG codons. Factors regulating 43S PIC binding. The assembly and function of eukaryotic PIC is enhanced by the formation of a multifunctional complex or MFC comprising of TC, EIF1, EIF5 and multi-subunit factor EIF3. EIF3 interacts with all other MFC components and the multiple sites on the 40S subunit to promote PIC assembly. It also recruits 43S PIC to the mRNA, possibly by interacting with mRNA as it emerges from the exit channel of 40S subunit. In case of mammals, EIF3 also forms a protein bridge to the mRNA by interacting with the EIF4G. The J subunit of EIF3 can bind near the A site and mRNA entry channel of the 40S subunit and obstructs mRNA binding in the absence of TC, thus ensures the TC loading precedes 40S binding to mRNA. mRNA containing secondary structure in the 5' UTR requires ATP and helicase activity to enhance binding of the 43S PIC at the cap and for subsequent scanning. The various subunits of EIF4 like F, E and G are bound together, and this binding helps to hold the lobes of EIF4A together and facilitate the ATP dependent helicase activity. EIF4G could interact with mRNA sequences both upstream and downstream of the nucleotides located in the decoding sites and could position EIF4A at the mRNA entry channel, unwinding secondary structure in advance of the scanning PIC. There are certain proteins which are bound to the 40S ribosomes that might influence the conformation of the mRNA entry channel to increase the processivity of scanning. Thus, the knockdown of such proteins inhibits translation initiation. Role of PABP. Poly A binding protein or PABP interacts with the EIF4G and mediates circularization of the mRNA by linking the cap and poly A tail in a closed loop. This stimulates the mRNA binding to the 43S PIC by the binding of EIF4F to the capped 5' end of mRNA. Formation of the closed loop facilitates reinitiation by post-termination ribosomes, and there is evidence that PABP interacts with polypeptide release factors ERF1 and ERF3. PABP also stimulates 60S subunit joining, and the poly A tail is required to block the inhibitory effects of two RNA helicases on subunit joining factor EIF5B. Factors regulating subunit joining. The EIF5B plays a major role in catalyzing the 60S subunit joining and second GTP hydrolysis reaction at the end of eukaryotic initiation pathway. EIF5B interacts with the extreme C terminus of EIF1A and stimulates both subunit joining and the GTP hydrolysis that triggers EIF5B release from the initiation complex. This interaction recruits EIF5B to 48S PIC for subunit joining. Meanwhile, the EIF5B promotes the release of EIF1A from the ATS initiation complex and open up A site for the first amino acyl tRNA. Thus, it is clear that EIF1A carries out the following functions: ternary complex recruitment, scanning, AUG codon selection, subunit joining. Let us now discuss about cap independent initiation. Usually, translation initiation starts with the recognition of the 5' cap structure or the 3' poly A tail for the binding of PIC by the initiation factors EIFs. In cap independent initiation, initiation starts at a place different from the cap structure, somewhere in the middle of the mRNA called internal ribosome entry sites or IRES. IRES is an RNA structure that directly recruits the 40S ribosomal subunits through a mechanism independent of the cap structure. It is found usually at the 5' UTR, but can occur in many different places in mRNA. It was first discovered in picorna virus in 1988 by Nathan Sorenberg and Eckhard Wimmer. This process is thus called internal initiation of translation. There are a number of cellular RNA binding proteins that are capable of enhancing internal initiation called IRES transacting factors or ITAFs. IRES elements interact with EIFs, ITAFs and 40S ribosomal subunits to provide a means of proper positioning of the initiation codon at the ribosomal P site without scanning from the 5' end. There are two types of IRES mediated translation. 1. Viral IRES. 2. Cellular IRES. Cellular IRES is less efficient when compared to viral IRES. Cellular IRES have two major physiological functions. 1. They support low levels of translation initiation for mRNAs with highly structured 5' UTRs under normal physiological conditions when cap dependent initiation is fully active due to its incompatibility with efficient scanning. 2. They support vigorous translation of cellular mRNA under conditions such as mitosis when cap dependent initiation is not active. Single nucleotide substitutions, small deletions or insertions can either reduce or enhance the activity of a particular IRES. IRES mediated translation initiation is a regulatory mechanism that facilitates cells to respond to and cope with various stress related circumstances. For example, ion control of translation by IREBP. The expression of genes ferritin which safely sequesters iron ions inside cells and transferrin which transports ion from the blood into the cell is regulated by the iron sensitive RNA binding protein. The iron response elements or IRE interact with IRE binding protein, the protein that recognizes and binds to the IREs. In ferritin gene, when there is high iron, the IREBP binds to the iron, so the IRE 5' UTR sequences are free for ribosome binding and allows the translation of ferritin. When the iron concentration is low, the IREBP is free to bind to IRE and prevents the synthesis of ferritin. In case of transferrin, the IRE on the transferrin receptor mRNA is found at the 3' UTR. When there is low intracellular iron, more transferrin is needed to bring iron into the cell. Under low concentrations of iron, IRBP is free to bind to IRE, forming a stabilized structure preventing the degradation of the poly A tail. IRBP binds to iron and fails to stabilize the IRE loop when concentrations of iron is high, leading to the degradation of transferrin receptor mRNA. Another prominent mechanism for blocking translation initiation is to reduce the activities of EIFs that stimulate methionine tRNAI recruitment to the 40S subunit. Although this might be expected to exert only a general inhibition of initiation, specialized mechanisms have evolved that allow certain mRNAs encoding transcription factors to be upregulated translationally under stress conditions where most translations are repressed. The EIF2 alpha kinases. EIF2 alpha kinase phosphorylates serine residue in the alpha subunit of EIF2. When EIF2 is phosphorylated, the GDP bound complex is stabilized and the exchange of GTP is inhibited. The exchange of GDP for GTP is mediated by EIF2B. When EIF2 is phosphorylated, alpha, beta and gamma subunit composed subcomplex of EIF2B binds more tightly, slowing the rate of nucleotide exchange and thus affects the rate of initiation. This type of regulation occurs under different conditions like endoplasmic reticulum stress, nutrient stress, viral infection and in case of erythrocytes limiting heme. For example, heme control of translation. In RBCs, if there is an insufficient level of heme, translation of globin mRNA must be restricted to generate functional hemoglobins. Thus, when heme levels are low, an eif2 alpha kinase is activated and limits translational initiation until there is adequate heme. The regulatory eif2 alpha kinase in this system is called the heme regulated inhibitor or HRI. HRI is also known as the heme controlled inhibitor HCI or heme controlled repressor HCR. Example, interferon control of translation. Translation regulation is also induced by virally infected cells in order to shut off the protein synthesis to prevent propagation of viruses. Interferons formed during virus attack like leukocyte or alpha IFs, fibroblast or beta IFs and lymphocyte or gamma IFs induces a specific kinase RNA dependent protein kinase that phosphorylates the alpha subunit of eif2, thereby shutting off translation. RNA instability. Regulated changes in mRNA stability can modulate the level of expression of genes. In the systems studied so far, the sequence determinants dictate mRNA instability rather than mRNA stability. mRNA instability is rather linked to ongoing translation. Both structure and sequence are important. The 5' and 3' terminal structures defend against degradation, and specific sequences within the mRNA may either provide targets to trigger degradation or may defend against degradation. Specific sequence elements within the mRNA may stabilize or destabilize it. In some cases, an mRNA can be stabilized by specifically inhibiting the function of a destabilizing element. mRNA decay pathway. The mRNA decay pathway is initiated by the removal of poly A tail. This disrupts the translation initiation complex and allows the degradative enzymes to access the 5' cap and remaining RNA body. The decay pathways are divided into 1. Deadenylation dependent pathway, 2. Deadenylation independent pathway, which is further subdivided into Endo ribonucleolytic decay and Nonsense-mediated decay or NMD. 3. RNAI dependent pathway. Let us now discuss each of these pathways. 1. Deadenylation dependent decay pathway. The first step in decay is shortening of poly A tail by deadenylase. Thus, the 5' cap is removed and the mRNA is attacked by 5' and 3' exonucleases. AU-rich element or ARE found in 3' UTR encodes certain proteins which affect mRNA stability, translation and subcellular localization. Interaction of the ARE with a destabilizing factor might promote rapid deadenylation by reducing the affinity of the poly A binding protein PABP for the poly A tail. In contrast, the stabilizing factor might enhance the binding of the PABP to the poly A tail, then blocking deadenylation. Second pathway is deadenylation independent decay pathway. In this, let us discuss about endo ribonucleolytic decay. The endo ribonuclease recognizes certain specific sequences within the transcript and cleave the mRNA internally, generating free 3' and 5' ends. These are easily accessible to exonucleases and get degraded. Some stabilizer proteins may block the binding of endo ribonucleases and prevent degradation of mRNA. Nonsense-mediated decay or NMD. In nonsense-mediated decay, mRNAs with premature stop codons are degraded. It prevents the accumulation of aberrant transcripts and truncated protein by ensuring rapid decay of the mRNAs. mRNA stability is regulated in response to developmental or differentiation cues, hormonal regulation, diurnal variation and stress. The third pathway is RNAI dependent decay pathway. RNAI or RNA interference was discovered by Craig C. Mello and Andrew Z. Fire. RNA interference or RNAI. Another mechanism for translational control that uses small RNA molecules is RNA interference or RNAI. The double-stranded molecules are cleaved by an endonuclease called dicer. This forms small fragments with overhanging single-stranded 3' ends called short interfering RNAs or siRNAs. This allows the short fragments to form a complex with several proteins such as RISC, that is RNA induced silencing complex. One strand of the siRNA is incorporated into the effector complex of RNAI, the RISC. The short RNA guides RISC to target mRNA and catalyzes an endonucleolytic cleavage, resulting in a post-transcriptional silencing of gene expression. Thus, the particular mRNA is no more available for translation. Translational control by microRNAs or miRNAs. The regulation of translation is not only by protein factors, but also by small RNA molecules of approximately 22 nucleotides in length that are known as microRNAs or miRNAs. miRNAs form stem loop structures and hybridize to 3' untranslated sequences of mRNA, thus blocking initiation of translation on those mRNA molecules. Since the target mRNA remains intact after miRNA binding, the miRNAs are believed to repress translation rather than stop translation by degrading the mRNA. miRNAs biochemically identical to another small RNA species known as small interfering RNA or siRNA. siRNAs are double-stranded RNA molecules of 21 up to 23 nucleotides in length, which mediate the degradation of mRNAs that show perfect complementarity to either of the siRNA strands. The miRNAs are transcribed as primary transcripts, which are then processed to yield pre-miRNAs and form stem loop structures. The pre-miRNAs are further processed by an enzyme called dicer to form mature miRNAs. Dicer also generates siRNAs from its precursors. The mature siRNAs then form an RNA-induced silencing complex or RISC that mediates the degradation of mRNAs that are complementary to the siRNA. miRNAs can behave as siRNAs, thus RISC might direct both mRNA degradation and translational silencing. Inhibitors of protein synthesis. There are certain factors which inhibits the translation or protein synthesis. Some well-known inhibitors of prokaryotic translation include streptomycin, erythromycin, tetracycline and chloramphenicol. Inhibitors of eukaryotic translation include cycloheximide and diphtheria and Pseudomonas toxins. Puromycin inhibits both prokaryotic and eukaryotic translation by binding to the A site. Peptidyl transferase attaches the peptide to puromycin and the peptide with puromycin attached at the C terminus is released, prematurely terminating chain growth. Certain antibiotics, for example chloramphenicol, inhibit mitochondrial protein synthesis but not cytoplasmic protein synthesis because mitochondrial ribosomes are similar to prokaryotic ribosomes. Let us now summarize what we have learnt in this session. Translation regulation can be used to maintain homeostasis in addition to modulating more permanent changes in cell physiology. Translation may be controlled by regulators that interact with mRNA. Most regulation occurs at the initiation stage of translation. Translation is controlled either by affecting the initiation factors, ribosomal subunits, the binding proteins or directly affecting the mRNA leading to its degradation. The mRNA instability can modulate the translation process. It provides flexibility in affecting changes in pattern of protein synthesis. mRNA instability is significant in controlling the rate of mRNA decay, thereby regulate the levels of cellular mRNA transcripts. Thus, unnecessary mRNAs are degraded or blocked from translation. RNA interference was a breakthrough in the regulation of translation in which a small RNA interferes with the mRNA transcripts, leading to its decay or inhibiting its translation. There are certain factors like antibiotics and toxins which inhibits translation. Here are a few questions for you to work out. 1. What are the major sites of translation regulation? 2. What is IRES? Explain. 3. How does a PABP function? 4. List out the initiation factors or EIFs involved in translation initiation. 5. Give some examples for inhibitors of protein synthesis. Here I can suggest a few books for your reference. Genes, Volume 8, written by Lewin B., published by Pearson Prentice Hall, publication 2004. Molecular Cell Biology, 5th edition, written by Lodish H., Burke A., Matsudaira P., Kaser C. A., Krieger M., Scott M. P., Sipersky L. and Darnell J. Published by Freeman W. H. and Company, 2003. Molecular Mechanisms of Translation Control by Fatima Gibor and Matthias W. Hentz. Here I can also suggest a few web links for your reference. www.studyblue.com, www.5prime.com, www.themedicalbiochemistrypage.org. Thank you for watching this program. Let's meet in the next session with a new topic. Until then, bye.